And so, as air's passaging through here, it's warming up to your body temperature. And as you breathe out through your nasal cavity, you'll be retaining that heat as the air's relieving. And so, your nasal cavity or your nasal compartment has that capability of preparing the air. And then we have another word here. This word, even though it starts with a CH, it's ko. Everything after sounds fine. Coena. This coena is the passageway from your nasal cavity into your pharynx, Now, pharynx is a general term to indicate the throat, what we've always just called the throat in our regular day-to-day lives. But depending on the compartment that it's adjacent to, for example, the nasal cavity, that's going to be referred to as the nasopharynx because it's behind the nasal cavity. Oropharynx is going to be the region adjacent to the oral cavity. And then finally, the laryngopharynx is going to be adjacent to our larynx, And so that's our pharynx. And this initial segment, I'm going to go ahead, you don't have to swap slides yet. You'll notice that if we look at an anterior view of this same structure, here at the very top, there is this lid-like structure called the epiglottis. And this epiglottis doesn't want anything but gases to flow in and out of the larynx. So when you're swallowing things, it ensures to shut. So it shuts this opening up, and then food will traverse down into the esophagus. And you also have that same feature up here, our soft palate. I know that in lab we talked about the hard palate, how we have the maxilla bone, and then we have the palatine bone, and they make up together your hard palate. If you again grab your tongue and you feel the roof of your mouth, that bony part of your mouth is your hard palate. And then that little soft part at the very end that if you're capable of tracing your tongue further back, that soft part is called your soft palate. And this will prevent anything that you're ingesting from going into your nasal cavity. And so it's like you have a protective layer covering this end and covering your airway. And the one that covers prevents the passage from food to your nasal cavity, soft palate, epiglottis, is going to be that cover or that protective opening or the structure that protects the opening entering into the larynx. And so with that. And so if this is referred to as our epiglottis, right? What does epi mean in Latin? Above. So we have a glottis, and this glottis is going to be this portion here that we're going to zoom in a little bit. But glottis is going to be underneath our epiglottis. And we have a better picture of that coming up. Anything I missed here? Yes. So I want to clarify on larynx and trachea, and that kind of transitions nice. You can change your slides. And so when you look at this model here, this is the anterior aspect of that same structure. Here you have your larynx, right? Your larynx. And here you can see your epiglottis. And what bone is this? Hyoid bone. And so we have a ligament named after the fact that it's going to be attached to that larynx. And this larynx has cartilages that we have to become aware of. The first one, thyroid cartilage. It's going to be a larger one named after the fact that there is a thyroid gland that sits roughly on both sides on this end, which we haven't really discussed yet, but it sits roughly around there, and it's named after that very fact. And then we have our cricoid cartilage, which is sort of like a mini version of that thyroid cartilage, right? It looks unique to the ones that remain that we haven't discussed, and it's clearly smaller than this one. So try to always train your eyes to learn the thyroid cartilage, cricoid cartilage, and then we get consistent tracheal rings, or these C rings, cartilage rings, which we'll talk about in a second. But right now, if we look at just at our larynx, it is made out of cartilage. That epiglottis will protect the glottis, and you all know this glottis as the voice box. And let's see if we can get a lateral perspective. And so I'm going to have to use two images here. If you don't have to swap back. And so when you look at this image here, glottis, you can think of to yourself as the opening, a passageway once something has entered the larynx and it's going to proceed down into the trachea. And that glottis exists within the larynx. And if we look at the size of these cartilage, you can see that this label maybe would be more complete if they actually wrote our thyroid cartilage on this one. And then the next one that follows are cricoid cartilage. Thyroid cartilage and cricoid cartilage. And if you look at the boundaries, this cricoid cartilage marks a boundary. All right, so everything that's cricoid cartilage and up is considered your larynx. The trachea begins where you start seeing those consistent tracheal rings. Those are going to be, that's going to be the beginning of the trachea. And so if we look at this image, an anterior aspect, we have our thyroid, our cricoid cartilage, and then right underneath, again, we have our trachea right underneath that cricoid cartilage. And just as a little last things to add here, our thyroid cartilage, what we've always just commonly as called the Adam's apple, is that thyroid cartilage. That thyroid cartilage is what makes up the Adam's apple. There are ligaments. The larynx job isn't just only to allow gas exchange or gases to go through, to go be at sites of exchange, to conduct air, but really it's also going to serve as a way or a place of attachment for our hyoid bone. And so we refer to that as the ligament, the median thyrohyoid ligament, because it connects to the thyroid cartilage and the hyoid bone, and hence the name ligament. This one you don't have to worry about, but this one you should know. This, on the exam, you can expect there'll be a labeling assignment to this one, so make sure that you can just identify this. And the good news is about lecture, when we hit this level, everything that you will be tested for in upcoming practicals really runs next to the lecture material. It's just happening at different times. I know that when you're learning skeleton and you're learning topics in lecture, it's a little tough, but everything that you're learning now, you will need in the future lab exams, which is really nice for all of us, right? We don't have to put or go double topics like we used to. And so, if we magic school bus our way into this larynx, right? We got really small, and then we went through that epiglottis. It led us through. What we would be seeing is our glottis. And you can see that the glottis is made out of the rima glottidis, which is going to be the opening, this opening here, marked by your brackets, and then your vocal folds. And so this is going to be what causes, like even right now, your ability for me to control the way that the wind is coming out of me is done by our glottis, right? I'm able to make sounds out of this. That's why it's typically referred to as your vocal cords. The vibrations, right now as I'm talking, these are vibrating to match whatever sound I'm trying to make, and so the vibrations produce those sound waves. This is a cartilage. I don't really care for you to know in lecture. In lab, it will come up again, so it was worth putting back in here. So you can put a little asterisk in here for later on. And then vestibular folds make up the remaining bit of this segment here. When you look at your vocal folds, if you were actually to sort of dry out this tissue, you would see that you would be left with just these two little twigs just sticking here, and this little connective tissue would sort of completely disappear. But this is a bit of connective tissue, a very thin layer of tissue that just serves to prevent foreign things from entering your glottis, which I always thought would just be the vocal folds preventing that, but yes, and the epiglottis together. All right, let's keep going. And so with that, we progress down now more inferior down the larynx, right? The larynx ended, right? We stopped. We don't see our thyroid cartilage. We don't see our cricoid cartilage, but we see our trachea. And so this trachea is going to have two main branches that stem from that one trachea. This break, even though it's not written on these notes, I want you to add it. This is going to be called your carina, that groove you see there, the carina here. And your trachea, if you think about it like upside down, can I flip this page upside down? Let's see, I think I can. Rotate this page. And so if you look at it, it kind of looks like a tree, right? Like when you look at it upside down, you have this trunk and then you have some branching. And that's all you're going to be learning about in the next few slides, is that as we progress down the trachea, we just get more narrow we start branching more, and then all the magic, right? All the gas exchange, all that oxygen and CO2 exchange and loading onto the capillaries and the blood vessels is all going to be occurring at the very end of our bronchial tree here. And so we start here at the trachea, and then we are going to begin to branch into both lungs. So let's go back and re-rotate this. And so before we get into it, something that might be easier to understand about branching is to understand that our lungs have lobes to them. You have a superior lobe and an inferior lobe on the left side. And the right side has 3 lobes, a superior. It also has an inferior, but it also has a middle. That's the only difference. And what's going to be occupied here at the cardiac notch? The heart. And so it's going to be on that left-hand side, the cardiac notch. These lobes can be distinguished by fissures. Fissures, as you're going to see, we're going to have to draw comparisons between fissures and sulci when we get into nervous. Fissures is a deep, deep indentation, a deep crack in the ground. If you think about like if an earthquake, like in a cartoon, like if there's an earthquake and the ground fragments and breaks apart, and then you can imagine people falling, rather, if we've all seen a movie or something like that occurring, that's a fissure, something that's really deep. A sulci will be much more smaller, but today we're only seeing fissures, and so you have 2 fissures to worry about. You have the horizontal fissure, which divides the superior lobe from the middle lobe. And then you have the oblique fissure, which separates the inferior lobe from the middle lobe and the superior lobe, if we follow it all the way across. And in the heart, recall that that pointed end was called the apex. So I told you that word was going to come back. Apex is the pointed ends at the superior aspect of the lungs. And then now the base is the opposite, right? Remember how the heart can be a little confusing because the base is at the superior portion of the heart and the apex was at the inferior portion. But when we look at the lungs, again, the superior portion will have the apex and then the base will be at the inferior aspect of it. We also give names to the borders or the boundaries of the lungs. This portion that's going to be on the front side of the body is going to be the anterior border. And then the portion that's at the bottom of the lungs is going to be the inferior border. And so these are some terms that we're going to come up with in a second, sort of sums up a few things. But one thing I just want to make sure that we are registering right now is that the right lung and the left lung have different amount of lobes. So make sure that we know that going into our exam. This will come up again in a second. All right. And so if we flip those lungs around and we look at the posterior, actually this is the medial aspect. This is where your heart is sitting. And the reason I know it's medial is what do we expect to be going into the lungs from the heart? What sends blood out of the heart and into the lungs? What vessels out of these 2? Which one? Pulmonary arteries. Are they carrying deoxygenated blood or oxygenated blood? One more time. Deoxygenated. Never forget the pulmonary arteries are carrying deoxygenated blood. Oh shoot, this is too thick. Let's make it smaller. Deoxygenated blood, or blood content with low oxygen levels, right? And the pulmonary veins will have what we're just going to easily just call oxygenated. And so remember that those blood cells that used up their oxygen and now they're carrying CO2, their goal is to go to the lungs and reload on that oxygen and let go of that CO2. And so the lungs need to provide a space for those vessels to enter the lungs. And so our pulmonary veins and our pulmonary arteries go in through an opening we refer to as the root of the lung. Root of the lung. And that's going to be this little opening here where you can see the large vessels that come from the heart and to the heart. They're going to enter through the root of the lung. The hilum refers to the actual segment where those vessels and even the bronchus, which we'll talk about in a second, that's where they invaginate the lung. They form this fold inside that root of the lung. And so the hilum is going to come up again because typically you're always going to see this infolding around blood vessels. We're going to see it come up again. And so hilum. So root of the lung is the entire opening. Hilum is that invagination caused by those things that penetrate into the lung. And again, you can even define it as the depression. All right, depression into the lung. And otherwise, we still have our lobes, and this one has 3 lobes. So what side is, oh, it's already in here for you, never mind. Superior lobe, middle lobe, and inferior lobe, right side. And then superior lobe and inferior lobe is only on the left lung. On the left lung only has those 2 lobes. And what muscle goes down here? It's kind of clued in here. What muscle goes down there? The diaphragm. It also marked the boundary between, where do the lungs and the heart sit? What cavity? The thoracic cavity. And what's the cavity underneath? The abdominal cavity. And then what's the other one that's all the way down inferior part of the body? The pelvic cavity. Nice. And all right, let's keep going. Anything I missed? No, let's keep going. And now to our larynx and our trachea. So again, here's our hyoid bone, our larynx to get us oriented. Questions? And so here our thyroid cartilage is going to be, again, that larger cartilage than the cricoid cartilage. And then finally we have our tracheal cartilages. These are going to be the ones that wrap around the trachea. And so they are sometimes called C-rings because sometimes they don't necessarily go all the way around. They sort of don't look like perfect rings. They look more like C-rings like this. And so sometimes you might see in some other textbooks they're referred to as C-rings. And the trachea, if we look at this level, it begins at C6, your 6th cervical bone, and your 5th thoracic bone. And how many cervical bones do we have? What time are we supposed to eat breakfast? 7. How many thoracic ones do we have? 12. So how many pairs of ribs do we have? 24. And so here you can see that from here, C5, let's write that note. From C5 to approximately T5, pardon, to C6. Mess up my numbers here. That's where our trachea can be found. Now, the branching system. This is a nice little flow chart that you might want to register before you even apply it to the images. It'll save you some time. But the trachea will branch into smaller segments as we progress. It's like a little, if you drew a line here, we're expecting the diameter of our airway, or the tube that is your bronchus and the bronchioles, this tube that air is going to pass through, gets smaller in diameter. And what did in Latin, what did we say that adding IOLES to anything meant? A smaller version of it. And where else did we see it in cardiovascular? What are the large vessels that send blood away from the heart? Arteries. And then what are the tinier ones as we're approaching the capillaries? Arterial. So you can see IOLES typically implies that something is getting smaller. So it's a play off of the word bronchi or bronchus. And let's clarify that. Bronchus. Oops. Bronchus. Bronchus is singular. Bronchi is plural. And so bronchioles is a play off of that word where bronchus or bronchi, and we're just saying smaller version of it. And so let's progress with that. And so if we start here at the carina that I mentioned, that the carina is this fold here, this band, this crossroad, right? Like you have this segment that goes into the left lung, you have one that goes into the right lung, and then that crossroad is considered the carina. The main bronchus is going to be the larger ones that are going to go and reach for the lung that they're targeting, right? If you're the right main bronchus, you're going to go to the right lung. If you're the left bronchus, you're going to go to the left lung. That one's pretty easy. But there's one little fun fact to add about the bronchus. The difference is that the one on the left is actually smaller. Let's see where I can write this down. Oops, here, larger. Larger than left. And so our main bronchus on the right side is going to be larger than the one on the left-hand side. And so, lobar. What do we think lobar is implying? The lobes, right? So the lobar bronchi are, their goal is to go into each single lobe. So how many lobes, a lobar bronchi, would you expect to have on the left lung? 2. And how many on the right side? 3, because lobar implies that they're all going to target an individual lobe, hence the name lobar bronchi. So you can see we're almost already halfway through. Now, the word segmental. The word segmental means to, when you segment something, means to like break it apart, to branch, to break apart into more branches, because we're going to see that word come up a little bit later down the line as well. And so we have now gone into a lobe through our lobar bronchus, right? Bronchus being singular. And then we begin to segment, right? We're seeing segmentation, more branching, because the overall goal for our bronchi is to reach the very, like to extend, have these extensions reach out and just use as much of the surface area or the volume of the lungs, in order to have all exchange sites for gases, which we've sort of learned about a little bit during connective tissue. But something that I want to address too, what do you notice about the features around the cartilages around those bronchi? What do we notice about, say, from this segment up top, this segment versus this segment? They look different, right? They're just different. Like these are rings and these are more like plates. And so the cartilage, excuse you, or pardon, what do you say? Bless you. And so this little segment here, this little segment here is your the little cartilage plates. So you can see that they're sort of the same thing as the tracheal rings, but they have a different conformation or different shape, and their role is similar in providing some rigidity and some structure to the bronchi. And so that's typically one of the biggest indications that we've already transitioned from a main bronchus to a lobar bronchus, is that we can see that we have those cartilage plates. Now, as we begin to segment, right, we're almost done with our little flow chart here. We're segmented. Now we finally are narrowing down in that little passageway. If we magic school bust our way into these bronchus, into these bronchi, into the tube, we're getting more and more narrow, more and more smaller as we progress. And then we're going to narrow, and we end up finally at what we refer to as our bronchioles. Now, the bronchioles are the slim portion. You'll also notice that it's the end of the cartilage. You don't really see cartilage around the bronchioles anymore. And as we're terminating, right, as we're reaching the very final destination, terminating, we finally reached our terminal bronchiole. The respiratory bronchiole is that final stage that gas needs to pass through in order to reach our alveoli and those capillaries in order to do gas exchange. And so the terminal bronchioles are where we're terminating. That's why I kind of like to remember it. And respiratory, the reason I kind of can remember it's not, first it's not respiratory versus terminal, is that I know that respiration, to me, I always think of it clearly more like internal respiration. And we'll just leave that. I'll clarify that in a second. And so with the terminal bronchiole comes before the respiratory bronchiole. Be careful, it's easy to mix these up. Respiratory bronchioles will be attached to the pulmonary lobules where the gas exchange will occur. Now, something to mention about our bronchioles is that when we look at our bronchioles, we sort of started touching on it, and you don't have to swap slides here. I'll do it for you. That as we were talking about the mucosa layer of things, or the lining right, of these cavities until we're going right here into the pharynx. And then as we go into the larynx, that the epithelial lining is transitioning from one thing to another. And so we saw in our nasal cavity, we had those columnar cells, pseudostratified columnar cells with goblet cells, cilia helping us move mucus through it. And as we transitioned into the pharynx, that we had stratified squamous epithelia, which specializes in what? Protection from what? Friction, exactly. And then because we're swallowing, ingesting food all the time, so we need to have a protective layer at all times in order to just sort of, or cells that are easy to just replace, right? We're always replacing these cells through mitosis. They're traveling towards the lumen space, the apical direction. And then we've made our way into the larynx. And in our larynx, we can see that we transition back into pseudostratified columnar epithelia, which creates that mucociliary apparatus, which again is going to be, we're going to produce mucus through the goblet cells. We're going to produce mucus through the mucus glands that's located in the connective tissue layer. They produce mucus into that lumen, and any trapped debris will be removed by the beating cilia and will move that mucus and all the trapped debris into your esophagus where you'll swallow it, or some of us might spit out a loogie, right? And one thing I wanted to ask, and I forgot as I was talking right now. I completely forgot. Oh, yes, no, that wasn't a question. And so this is why if you smoke things like cigarettes or you just smoke anything combustion that puts a combustion in you for a long time, like what's an easier way of saying this? Whenever you smoke cigarettes, you paralyze the hairs on these cilia. And so that's why those individuals that have typically who are chronic smokers have a dysfunctioning mucociliary escalator. And so they rely on their tracheal muscles that surround your trachea and your larynx in order to project that mucus out because their mucociliary apparatus is not functioning very well. And so with that, we've now transitioned into what looks like our segmental bronchi, right? Our segmental ones. We segmented, and what we see in there is cuboidal epithelia. And then finally, when we reach these very pulmonary lobules, we're going to see that we have simple squamous. And what was simple squamous great at? Gas exchange. Awesome. And doesn't it make sense that we're talking about gas exchange now, right? And simple squamous. And so if we magic school bust our way into the bronchioles and into the bronchi, again, this would be what the passageway looks like. It's not a perfect little circle. And you can see that this is the epithelial layer that we're talking about. And underneath the epithelial layer, what's that usually indicated by a line underneath the epithelial tissue? What was it? The basement membrane. And what's underneath the basement membrane? What's the family of tissues? Connective tissue. And then now, these muscles, oh, dang it, it's written there. And so smooth muscle is going to be that next layer. And as we transition into digestive and urinary, we're going to get really into layers. And so what was that first layer? Anybody remember what it's called? What do we call it? The open space is the lumen. What do we call the actual lining? We gave it an M. It starts with an M. Anybody remember? It produces mucus, and it's named after that fact. Mucosa. So this will be your mucosa layer because the mucus is going to be on that surface. And so brings me to my next point now, is that you have some vocab words here, bronchodilation and bronchoconstriction. Dilation is when the diameter increases, right? If you have this is your opening. If you mean by increase, it gets larger. Increases, dilation. This word will come back to haunt us. Dilation means to increase in diameter, and therefore it can increase airflow. So if you take a bronchodilator, you kind of understand what's going on, right? It's increasing your airflow by allowing the relaxation of those muscles, or rather just the increase of the diameter of those muscles. This is something that's here, and I don't want it to scare you. We haven't gone over nervous, so you can go ahead and neglect it, but I want your notes to be complete and they'll make sense after we go over nervous. But otherwise, this is something you don't need to know just yet. And so bronchoconstriction now is going to be the opposite, where we decrease that airflow because we are constricting that flow, that passageway. We are making it smaller. And so when we look at this lumen, this space inside, when we say something is going to constrict, again, oops, sorry, wrong thing. When we're there, they are constricting, right? What's doing it is going to be the smooth muscle. To make this figure a little bit more complete, right? You want to have the smooth muscle sort of lined out, smooth muscle. And this would be vasoconstriction, vasoconstriction, vasoconstriction. And then dilation, these muscles need to relax, and so you will get, they would increase in diameter. So I'm going to write vasodilation. Vasodilation. Sorry, my writing on this is getting worse, I feel. I'm just going to go ahead and write separately. All right, to increase that airflow. And so some things, like when you have extreme vasoconstriction, we refer to this as asthma. And this can be caused by a few triggers. This can be caused by allergies, hypersensitivity, and a few other things. But we usually refer to anything that causes extreme bronchoconstriction as triggers. You might see this again, triggers. Ask away.
So, like for an allergic reaction, when your throat comes off, is that the same type of thing?
Like when you're going into anaphylactic shock? I'm going to look that up for you because I'm drawing a blank on it and I don't want to say something wrong. And I feel like that's so important to know. And I should know it, but I don't remember. I'm going to have to put an IOU for that, but that's a good question, and I will find that answer. I have this little activity that we're going to do in a split second, and that's when I'll figure it out. But I really would like to know the answer to that, too. All right, so with that, let's keep going. And so we're almost at the end of our slide set here. And the last thing we need to do is understand the gas exchange that's occurring at the capillaries. And so if we look now where we left off at our segmental bronchi and our terminal bronchioles, and finally the respiratory bronchioles right before we hit the alveoli. And so we refer to that terminal bronchiole and the associated alveoli as a bronchopulmonary segment. A bronchopulmonary segment. If we zoom in on our alveoli, right? This is the image that we're seeing here. It's a zoomed in image of our alveoli. And so if we start at the very beginning here, if we're a little gas molecule, whether it's oxygen or CO2, right? We are not traveling through this blue one. What is the blue probably indicating? What does blue typically indicate when we talk about vessels? Veins. And what does red indicate? But in the lungs and in the after the heart, major heart vessels, what's the difference? Yes, the blue ones. And why is that? Why is the pulmonary artery, why is this painted in blue and not red? Because it's deoxygenated. And so now our deoxygenated blood cells, right, they're traveling through these blood vessels, and finally they found their associated capillary. And now when you breathe in, right, they are going to associate with these little sacs here. And so if we look at this image, if we remove the capillaries and all those vessels, we see the last ending, or that ending, like if we're traveling down through the lumen, we're oxygen, we're traveling. We made it through our respiratory, bronchial, and then finally we enter what we refer to as our alveolar duct, which is like the last passageway that's going to allow oxygen into this entire sac, right? And so alveoli is referencing every single one of these globes here, every single one of them. Alveolar sacs is referring to a few of those. So alveoli is an individual globe, and then the alveolar sac refers to a bundle of them. And so the air, or oxygen, will flow in through the alveolar duct, and we'll zoom in on exactly what's going on here in a second. But otherwise, some other structures to look at is, if you think about now, every single one of these terminal bronchioles will lead to a respiratory bronchiole, and then at the very ends of all of them, you will have these bronchopulmonary segments and their alveoli. And all of them will lead gases into these segments to be exchanged at the capillaries. And so something to add before we actually break down the capillaries on this next slide and the alveoli is, we talked about the pericardial cavity. What was the layer that's associated directly on the heart called? Visceral. And what's the layer associated on the outside of that pericardial cavity? The outer layer? The parietal. So recall that we use those words for the pleural cavity as well. So you have a visceral pleura associated with the lungs itself. And then you're going to have a parietal pleura that's associated with the thoracic cavity, the outer layer, right? The outside. And then the inside, what's going to be inside? Serous fluid. And what's serous fluid good for? Lubrication. Very nice. And so, as we progress, let's see, anything that I missed here? And that's really it. All right. Now, exchange. What's happening? So if we zoom in on these alveoli, right now we're zooming in exactly to see what exactly they're made of. And the good news is that you already do know what it is. Every single one of these is a simple squamous cell. A simple squamous cell. You can see a lateral perspective of the cell. This is probably what you were more used to seeing, that if you look at this aspect here, I'm going to try to draw it in. Here is a little bit of that cell and the plasma membrane boundary there. And then another one here. And then another one here. But now you're seeing it from a 3D perspective. You were only ever seeing like a single layer of the cells, but these alveoli are like balloons made out of those thin, tiny little cells. And so they make these tiny alveoli that, again, require oxygen. And now we're looking between, embedded within the connective tissue and adjacent to the simple squamous, we see our capillaries here. These are our capillaries. This is where those red blood cells will never escape from. They're traveling through here, right? They're traveling through here, and this is where the exchange occurs. And what was the name of the simple squamous cells that are inside of blood vessels? Endothelia. And so you can see that you have a simple squamous tissue here, you have a simple squamous tissue here, and they are allowing for the passage of gas. They're great for that very thing. So we're seeing their main superpower, right? That's the simple squamous was good at gas exchange, and endothelia is just another name for a simple squamous within the blood vessels. Again, great at gas exchange. And so we'll talk about that a little bit more. And so we refer to those simple squamous. We don't call them simple squamous. We actually call them pneumocytes, right? What does cytes mean in Latin? Cells. And what typically is pneumo probably referring to? Something in the lungs, right? Pneumo. And so pneumocytes type 1 is going to be the simple squamous cells. Let's make a note of that. Simple squamous. I'm just going to put that S and S. And then we have our pneumocytes number 2. These are going to be responsible for producing this type of fluid that coats the inside of the alveoli. Like on top of this, even though that we're looking at the individual simple squamous cell, you have to imagine to yourself that each one is coated with a fluid, and this fluid is called surfactant. Surfactant, as written on your notes, with surfactant right here, surfactant. And so this fluid, it allows for some things. I'm going to just try to describe this as easily as possible, because what you have to imagine is that an implosion, right? If something expands, it's easy to understand when something's expanding. But what about shriveling into itself? That's what surfactant is for, is to prevent this shriveling, this sort of I don't want to say implosion, but this sort of to the integrity of the overall alveoli is maintained in a nice and spherical, and without surfactant, you can expect that it's just sort of going to collapse on itself. And so collapse sort of becomes the best word to use there, collapse. And so that's the role of surfactant, to prevent collapse. And then macrophages. Macrophages, do we remember what they do? What were these? What family of blood cells are they from? They are white blood cells, and yes? Yes, they definitely can break down red blood cells, absolutely. Yes. And what did they do with that iron? What sends it back to the bones? What carries the iron back to the bones? Transferrin, very good. And then that heme group produces those other wastes that are going to go to the liver. Do you remember what they are? It starts with B. Good. That's very good. And then so now, as we go through this segment now what's next? All we got to finish up now is the blood-air barrier. Now, this image is sort of like a sandwich or a reflection of the view of this very thing here. If I zoom in an exaggerated manner, you're seeing the inside of the capillary, and then you're seeing that endothelia, and then you're seeing the pneumocytes 1, the simple squamous on the alveoli. And so that's what we're seeing here. The capillary lumen and then the alveolar airspace. You can see that this is the thin layer of the pneumocytes 1. They create the alveolar cell layer. And so these pneumocytes are simple squamous, great for gas exchange. And also the capillary lumen has our endothelia, which again also is great for gas exchange. And there's just a basement membrane that fuses them. And what typically has always been our formula when we talk about tissues? What tissue do we usually encounter where there's a lumen? What's the first tissue? The family of tissues, type of tissue? Epithelial. And then what's that boundary between basement membrane and underneath? And so you can see here we have a unique circumstance where you have two epithelial tissues and there's no connective tissue. Our formula of after the basement membrane, then connective tissue follows. Now we're not seeing that, so you can see this is referred to as a fused basement membrane, right? Because endothelia and the alveolar epithelia, or pneumocytes number 1, right? Pneumocytes type 1, they're going to have basement membranes fused together. There's no connective tissue in between them. Oh, sorry, wrong thing. There we go. And so with that. Oh, anyways, back to macrophages before I got distracted with that amazing performance by Jim. And then, so now, when we look at alveolar, right, these macrophages, what they're doing is that they're defending you against any possible pathogens and removing cell debris. And that's going to be these little purple ones on my nose. I know your notes don't have color, but these here. All right? And with that, anything I'm missing? Nope. All right. And to finish up our respiration, and then we're going to spend the last bit of class just showing you how to use that little study sheet that I made for you for cardiovascular. As I know that the heart is a little confusing. And so, last two topics to address on this packet. We have our external and internal respiration. External respiration deals with what exactly it sounds like, the external environment. So you can see our definition, exchange of gases between the blood, lungs, and external environment. All right. This gas diffusion occurs across our blood-air barrier that we just discussed between the alveolar air and the alveolar capillaries. In other words, it's the gas exchange occurring at this site. At this site. That's the limit of it. So if we look at the full, let me get a full picture here of a full body here. So if we think about everything that's the conducting through here, right? If we have air that's coming down, going down, 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 this is the outside world, right? The external environment. You breathe in, nasal cavity, pharynx. We go into our larynx. We go into our trachea. We go into our main bronchus. We go into our lobar bronchus. We go into our segmental bronchus. Then we go into our terminal bronchioles, and then the respiratory bronchioles, and then we exchange. That's the limit of external respiration. That's the limit we're talking about. Now, what is that versus internal? What cells in your body typically need oxygen? Almost every single cell in your blood needs oxygen. So that's what internal respiration is. It's when we actually exchange the gases with the tissues. So let me bring up an image that might make that a little bit easier to understand without all my stammering. And when we talked about capillaries, right, we talked about that these red blood cells, they're going to be traveling through here, and they're going to have oxygen, and then they're going to deposit their gas, right? They're going to send their gases or any nutrients into the interstitial fluid. And so this tissue, it could be any organ that's going to serve, or a capillary is going to serve, whether it's a muscle, whether it's your alveoli, maybe not your alveoli, but your any organ that you have in your body, anything that has capillaries, there will be an exchange site of gases. When the gas is being exchanged with the target tissue, then that's referred to as your internal respiration. In other words, after the lungs, right, that blood that's oxygenated will travel to, say, this muscle. And then it's going to provide some gas exchange to that muscle. That's internal respiration. Internal respiration. All right, that's the difference between the two. And otherwise. And the last thing to really add is to make sure that when you're looking at gases, you're thinking about the gases in general, that you are always thinking about oxygen has this directionality, right? That it's going into the alveoli, the oxygen, and CO2 is going to be diffusing outwards into the alveolar space. In other words, with this picture would be better, that CO2 will be usually leaving the capillaries, and then you breathe it out, you exhale it. And then oxygen will be traveling from the alveolar airspace into the capillary lumens. You just want to be a little fluent with that. And ventilation, the word ventilation means to air movement within and out of the lungs. And I think we'll draw the line there. All right. So this wraps up the material for next week's exam on Thursday. And so anything else? That's really it. All right. And so what to know? Let's go through your packets very quickly and just kind of how to prepare. Write some notes at the headings and sort of what I'm going to be focusing on and how to prepare. And so I know that a drop season is now, right? Like, you're deciding whether, some of you may be deciding whether it's time to withdraw or stick around for it. So we do the we drop 2 exams in this class. We do a lot of little extra credit in the very beginning to provide a little bit of a buffer. And that way you can see if I'm the professor for you, if you want to keep on going. And so it lets you make mistakes, right? Because this class is pretty intensive and we're not really sure how intensive it is until a little bit too late, right? And so all I'm going to say is that there's still a lot of points left in the class. So if you are staying in the class, which I really hope you do, there's still a lot of points and a lot to come back from. So your grades are complete right now, except for the extra credit that we did in lab today that hasn't been added. And the last thing I wanted to add about that. I guess that's really it. All right, so. Oh, yes, and tips. Oh, yeah, so also I had a lot of students, right? This is the week that I usually talk or reach out to a few of you maybe that are struggling, or you're reaching out to me. And so some big tips that I've noticed. You have that ebook, that's a cheap option, or the textbook. And one way that I would really recommend is these notes are from the publisher, and sometimes the reading is even easier than understanding it in a bullet point form. And so I would really love it if you have these notes as companion notes, that and you look through your book and you're looking at each figure, and whatever page you see this figure on, you read that segment. And what I love about this book, this isn't one of the most dense books. This is a very nice book. If it's just one page, has entire chapters onto one page, you flip it and you already finished one complete chapter. And in previous, not chapters, but like segments. And in previous books, there'll be like 10 or 15 pages just for that part one. This is one of the best books, and I promise you, it's not as hard or excruciating reading. And it will prepare you well for these exams. And so look for the figures. These are figures ripped directly off of that book. And these notes are directly summarizing what the readings are. And so you can find all of these notes and read everything about it. That's one of the best ways, I think, personally, to prepare. And yeah, I think that's one of my biggest tips. And the last one for lab, too. The next one for lab, like we do the extra credit assignments, right? We did the bone ones, like I say, okay, learn all of these today. At the end of class, we're going to do an extra credit. I'm going to ask you questions, and you better know it by memory, right? It's just trying to get you to just start the study process. And I want you to think about that time period. It's about, let's say after I'm done talking and we give like an hour, right? An hour for muscles today, about an hour and a half. And think about your experience when you went through that interview process with me, like how clumsy it was, right? It took you an hour and a half to do, if we think about that list, right, it has approximately 40 terms on that page, maybe less, the 30. And so an hour and a half to not even really master 30 to 35 terms. An hour and a half, so understand that cramming is crazy for this class. It's crazy. You spent an hour and a half and you didn't even master that one page. I'm not trying to be mean or anything, just to put things into perspective. And so imagine that you're going to need an hour and a half and maybe another remaining hour to just finish that page masterfully, to be able to ID them and to be able to give me the action for them. And now think about that first page that we did last week. You might need the same amount of time for that page. So you're looking easily about 6 hours of study to master, where you get nothing wrong, right? Master. You're going to need at least 6 hours to be studying for those exams. That's why I get so mad at you guys when you guys, I don't tell you, but, I get mad at you guys because I know you can get an A, and it's just a time thing. It's a time issue. It's just about putting the terms in your head and making the time for it. And so I think just as a segue or going into the future, that's one thing. Use the lab time. I really believe you can get a lot of it done in lab where you're not spending your Saturdays, Sundays. You can use some of that time for some downtime, and you can use the lab time a little bit more productively. And that's why my assignments are just study this, and then I'm going to test you after, because it's just to get that study process out of the way. But you're all capable of getting an A in this class. And all right. So those are the little things that came up, I think, talking to a few of you that I think would benefit everybody. And that's really it. Yes. And and last thing, big warning. That book as well covers anatomy and physiology. That's why I say look for the figures that you have in my notes and then do the reading on it. Don't read excessively. Look through my notes first and then find what you need, because that can be detrimental. That happened to a few students in previous semesters that I think might affect some of you as well. All right, so with that, have your blood packet out. So I think just normally everything that's in bold is important, always knowing our functions. This is a nice little summary table of your functions. And over here is the reading on those functions, just a little bit of facts that we went over. Make sure that we know them. This image is stolen or kind of ripped off of this entire image. Know this image. Know absolutely everything on this image. All right? We need to know our percentages of plasma. We need to know our percentages of formed elements. We need to know what are the components of plasma. And we also have to know the percentages of those components. Okay? We need to know examples of electrolytes. We need to know organic nutrients. What are our organic nutrients that we always just completely repeat over and over again? Carbohydrates, proteins, lipids, and nucleic acids, right? Organic nutrients. And what's an example? And no examples of our organic wastes, right? I forget that the notes already show it. And here, the plasma proteins, you make sure that we know all our plasma proteins and their functions. All right, and know our lineages, right? We have lymphoid stem cells. We have to know what cells arise, all right, or differentiate out of these stem cell populations. Lymphoid stem cells, and then create our lymphocytes, and then the rest of them are made of myeloid stem cells, right? Made for or differentiate from myeloid stem cells. We have to know the functions of every single one of our white blood cells, right? We have to know the quantity. We have to know which ones are the most abundant and which ones are the least abundant. Anybody remember our acronym for that very thing? Yes, never let monkeys eat bananas, right? Never let monkeys eat bananas. And then we have to know our categorization, right? So we have to know which ones are granular, which ones are agranular. And now going back to red blood cells. Everything in bold is important. Everything in bold is important. And everything in bold, make sure we understand. Nothing too difficult there. And then we had our blood tube packet. And so this was on erythropoiesis. This was a specific pathway between our myeloid stem cell becoming our erythroblast, becoming a reticulocyte. And as Jim masterfully pointed out, here we have our macrophages, right? They're capable of digesting old red blood cells, breaking their components apart for either recycling or sending them somewhere else. So, right, our heme groups and the iron from that heme group will be delivered by transferrin back to our red marrow in order to be reutilized to produce new hemoglobin. So we have to know this entire pathway. This entire figure you must know. All right? And this is just pulling off the last little segment, just because when we talked about it, we broke off this little segment just so it's not so overwhelming. But it's really what happens to biliverdin as it gets converted to bilirubin. It gets transferred to the liver, right, carried by albumin. And then what happens in the large intestine and how bacteria turn into urobilins and the color of feces. We talked about how that plays a role in that and how the kidneys play a role in the removal of urobilins as well. Urobilins. And so pretty much that's all that packet really had. And blood typing. Do not forget, we need to understand our blood typing. I might give you a scenario type. AB person might have, we have a patient who has type AB blood. Who can he receive blood from? Be able to answer that for every single blood type. If you're type A, who can you receive blood from? Who can you donate blood to? And so be able to do that for every single one of these segments, or every single one of the blood types. Be able to even address, I guess it all applies to the same thing. And be able to describe what's going to happen if you donated the incorrect blood type to somebody, what occurs, right? And don't forget that whenever we talked about like A positive, B positive, we were talking about the antigen that we found in rhesus monkeys, usually just called Rh antigen. And so the positive, right, if you're O positive, you also have this Rh antigen. If you're negative, that means you do not have it. Otherwise, not really much to add here. All right. I know, we're almost done. And then we have our cardiovascular. So this little friendly handout I gave you just sort of pulls out all the terms that you should just know in general aside from this packet. They're in the packet, but they're listed there for you so you can test yourself without any notes. Because that's the other thing that scares me about some of the way that some of you study is that you look at your notes, you look at it a bunch of times, you can say it while you're looking at your notes almost perfectly, but then the second the notes go away, there's a lot of fumbling, right? And then so you always want to put yourself in the perspective that, you know, you have to impress the person in front of you and try to just use a blank image like what you see at the very back here, like a little labeling assignment, that you want to just be able to address these or be able to cover these up, and you really want to practice labeling. And this second page. And download it into your laptop. Might have to connect to my laptop for this one. And yes, I will have to connect to my laptop. Oh.